Right, wonderful wax caps of Wales. Gareth Griffith, Professor Gareth Griffith, is a professor of fungal ecology at Aberystwyth University, and also the chair of mycology in the Department of Life Sciences there. But his specialism is grassland fungi, the ecology of grassland fungi, and with research interests going to fungal taxonomy and DNA barcoding, which I think is going to mention tonight as well. He's also quite well known and contributes to many periodicals to do with ecology and pathology, fungal ecology and plant pathology, not just here, but also in America, on the Conservation Committee of the Mycological Society of America, and well known in the UK for his expert knowledge and experts information about fungal systems, full stop, really. So he's an extraordinarily well qualified man to talk about this, and I'm really grateful that he's managed to give up his time to do just that. So I'm going to hand over to you now, Gareth, if that's okay, and thank you very much again.
Oh, thank you very much for a very warm, kind introduction and a warm welcome, and thank you for the invitation to speak as well. I'll just get my talk slide up, just make sure that's going to work okay. After Cwyr Campus Cymru, one of the wonderful wax caps of Wales. That was an appropriate title for the audience I have this evening. It's got to work out what these things are not. There we go. I thought I'd start a bit of historical context to wax caps. So I've got this nice picture from a book that's 250 years old that shows one of the earlier wax caps to be named, and this still has the same name. I think originally it was called Agaricus conicaeus, as you can see in the text there, but that's because Linnaeus didn't like mushrooms very much, and he called any gilled fungus he called Agaricus. And then Jakob Schaefer, who's a priest in Regensburg, so you find just like Gilbert White and lots of other people, it was the clerics who did a lot of the early botany and mycology and identifying these species and giving them these Latin binomial names that Linnaeus had invented. So here's the first one. I think any of you, I'm assuming that most of you know what a wax cap is. I shouldn't take that for granted. And you've probably seen this lovely red one. So this is the commonest of the bright red ones, Hygocybe. If you find this in a grassland, that's a good sign. I think any red wax caps is always a sign of a fairly good quality ancient grassland. Anyhow, this is the earliest one, 1774. But at about the same time in Wales, there was another priest or minister. This is Hugh Davis, who was the, what's he call it? Curate, I think is the right word, of Aberystwyth, which is not that far from where you all live. I'm sure that there might be somebody in the audience who is actually living in Aber, so you'll know the nice vicarage with the very ornate chimneys in the village. So that's where he used to live. But he was actually from Anglesey, and he used to. There wasn't a bridge at that time when he lived. He was sort of 1737 to 1821 were his dates. So there wasn't a bridge to get to Anglesey. So when he wanted to go and visit his family, he'd have to take that path across Traeth Laban, which is, I can't believe that people used to do that. If you look at it, it looks a tremendously risky thing to do. Anyway, he published. I think he was quite a shy person. He was most famous for the person who used to promote his name most was Thomas Pennant, who's another famous North Wales naturalist, and the father of travel writing and the top zoologist in his time, who was friends with Linnaeus and so forth. So Thomas Pennant could only do what he did because he had all these friends who were also good naturalists who used to feed him information, and Hugh Davis was one of them. But Hugh Davis was eventually did publish a book when he was quite old, 1813, so he was presumably long retired then. He called it Welsh Botanology, but in fact it's actually the plants of Anglesey and nowhere else in Wales. So sadly, we know that he was, I'm sure he would have known about the plants of Conwy, because we know he crossed to go and see Thomas Pennant. He used to have to cross the river, and again there wasn't a bridge there, unless you went all the way to Llanrwst. Apparently there was a rope that you had to hold as you took your horse over, that he wrote about in one of his letters to Thomas Pennant. So doing botanizing or looking for fungi was a much riskier business in those days than it is now. Sadly, he didn't write anything about all the fungi he found in Gwynedd, only the ones in Anglesey. It's only a few pages and most of his focus was on plants. But you can see I've just screenshotted the relevant page here, or the relevant bit of text. So it's got agaricus, which is what all the guild fungi were called, as I mentioned, and the Welsh names, cawsllyfant, we still use that. Bwyd y barcud, that's a bit strange, or fallen out of use, and bwyd y llyllon, which I guess would translate as fairy food. That kind of makes sense. Bwyd y barcud, kite food, is a bit weird. I don't know why I don't know how they came up with that name. I think he must have just asked local people what they call different mushrooms, because most fungi people just used to give them one name, mushrooms or madarch or whatever. Anyway, he did notice wax caps because he talks about Agaricus citicinus and a number of beautiful varieties. So from that, I reckon the green one is Agaricus citicinus, the parrot wax cap, and the beautiful varieties, I guess, would be the other colours of wax cap that he didn't mention much in his book. So I think he just classified them as one species. Because he wrote this book, as you can see, in Latin, English and Welsh, this book ended up being filed by librarians under Celtic literature or Welsh literature. So when people in about 1900 or just towards 1900 were going through all the books, all the botanical books, to make sure which species had been named by two different people and so forth, and had they missed any species, they never got sight of this. It was only, I think, about the 1960s that a guy called R.W.G. Dennis, who was a very famous mycologist at Kew, discovered the book and brought it to wider attention. And there's one species here, he talks about the Agaricus dilleni here, which we're not quite sure. We have to kind of have a guess about this thing because there isn't the species anything dilleni. Dillenius was the guy who taught biology at Oxford when Hugh Davis was a student at Oxford. But he talks about where he found it. Here you can see the old pastures between Bodgylchad and Cefn Coch. That's near Llansadwrn in Anglesey. And I've gone wandering there, but all the pastures, unfortunately, the ancient pastures that were there back in the 1700s have all been ploughed and reseeded. I actually worked out there was a second cousin of mine who lived at Cefn Coch, and he's quite an intensive farmer. So I didn't even bother going to ask him if he'd seen any wax caps. And I'm pretty sure that this fungus that he has found here is no longer there. But I reckon that this is probably Entoloma bloxamii, which is a very rare fungus. Unfortunately, he didn't keep a specimen, which is what happens these days. That's my best guess, and I've discussed with other people because he talks about another book here, Withering, and there's a bit more description there. So sadly, because this book got misfiled, it's called Bloxamii after the Reverend Bloxham. So another vicar got the credit for discovering that species, not Hugh Davis. So that's a pity for Hugh Davis. Anyway, so move on a little bit to the present day. I guess I could give some explanation for why I started studying wax caps. Well, one of the reasons is that if you're doing mycology or fungal ecology in Wales, and you look at all the other fungal ecologists in Europe, they're mostly looking at woodland habitats, forest fungi and this type of thing, the ectomycorrhizal fungi that are associated with trees and so forth. There's rather meager pickings in Wales with woodland, as you probably noticed. And here you can see that we've got a huge amount of our land area, bigger than even in Scotland, is rough grasses and rough grazing. So, and I also realised that hardly anybody was studying the fungi you find in grasslands. I mentioned that word ancient grasslands just earlier. An ancient grassland I'd call one that hasn't been ploughed. It may have been abused in other ways, heavily grazed, maybe some, within reason, some fertilizer addition. But these ancient grasslands is where you're going to find the wax caps. And in the lowlands, more than 90%, probably more like 97% of the lowland grasslands, the ancient grasslands that weren't ploughed, have been lost since the Second World War. In Wales, the level of loss is probably lower in these upland grasslands because it's difficult to get a tractor up to the top of these hills and on the steep slopes. So it's the topography that's really protected the grasslands in Wales. So what do I mean by ancient grassland? Another term that's more widely used probably is waxcap grassland. I'd equate the two. I think the best indicator of an ancient grassland would be the presence of wax caps, because you quite often find fields that have been very heavily grazed by sheep for many years, and all the interesting plants have been eaten out of the sward, but the soil is essentially untouched. So if you see the wax caps coming up, you know that that's an undisturbed grassland, and if you change the management, I think reasonably quickly you'd have your botanical diversity would come back in again. So here you can see this is a view from towards, say, Pwllheli up in the top there, from the side of Tre'r, and you can see this grassland here has lots of wax caps in it, whereas these grasslands here have all been ploughed and reseeded with ryegrass, and you'd find very few fungi, very few wax caps, even the field mushroom Agaricus campestris. You'd struggle to find that there. These fields get ploughed and reseeded every 5 years, and they get a lot of fertilizer, often more than a wheat field would get. So we've got a big problem across the UK with excessive use of synthetic nitrogen fertilizers. But anyway, in these untouched grasslands, and presumably this hasn't been touched because it's on a slight slope, and you always worry when you go past these nice fields and wonder whether the farmer might suddenly decide he's going to plough and reseed the grassland just to increase his productivity. Anyhow, if you look carefully in these grasslands, you don't just find wax caps, which I think you're all familiar with, but you also find fairy clubs, which is on the left here. In the middle, you've got the pink gills, the entelomas, like the one Bloxamii I mentioned earlier, and the earth tongues, geoglossums, and the cracked caps, dermaloma species. So there is this acronym CHEG-D, which is the first letter of the family name of each of these groups and the genus name here. It's a really ugly name, CHEG-D. I'll blame my late friend Maurice Rotherrow for that. But it's a useful term as an ecological grouping. These fungi tend to occur in the same types of habitat, and the more we look at them, we find other similarities between them. They're all, you can see that there's 3 groups of mushroom forming fungi. These fairy clubs and wax caps are more closely related to each other than wax caps are to pink gills or dermalomas, which might surprise some of you, but it's just basically the ancestor of the fairy clubs was probably a mushroom with gills, and it's taken an evolutionary path that's given us this rather strange fruit body morphology here. Presumably, we don't really know why it happened. Since the only purpose of the mushroom or the fruit body is to spread the spores, we presume that these fairy clubs spread their spores in some other way than spreading them into the air, because a mushroom is a really perfectly adapted structure for spreading spores. So the billions and billions of spores each mushroom produced are very effectively spread by a mushroom. Something else is doing the dispersal of the spores of the fairy clubs, but we don't yet know. And the last group I have mentioned, the geoglossums, the earth tongues, these are in a different group of fungi, the Ascomycetes, the spore shooters. So they're quite distinct from the other 4 groups. And if you take one of the, if you find an earth tongue, if you put it in a petri dish or some clear container that will keep the humidity high, and if you leave it overnight, you'll see there'll be a little cloud of spores on the clear surface of the dish, because it's shooting its spores out. And the spores, if you look down a microscope, are amazing. They're about 1/10 of a millimeter long. They're like javelins. I don't know how many. I don't know how many of you own microscopes. You can see a lot more about fungi if you use a microscope. But I know a microscope is a bit daunting. So on the left here, you've got a map showing the conservation status of the citrine wax cap, Hygroscibes citrinovirens. And you can see it's mostly in the westerly areas of the island of Britain here, where the upland grassland is, and there's higher levels of rainfall. So it's difficult to know whether it's less common in the east because the climate is less suitable, or because the agriculture is more intensive in that area. So nobody can have avoided the news, or the constant news and the depressing news about the sort of, what's it called, the sixth global extinction that we are causing, our species is causing on the planet. So in millions of years to come, if people look at the geological record, they'll see an awful lot of species will have gone extinct about now, and that's caused by us, which is a rather depressing thought, and it doesn't seem to get that much better. It just seems to accelerate because it's never the top priority. But you have organizations like the International Union for the Conservation of Nature, and people try and assess the extinction risk of different species. And I've got a picture here of a red kite. So usually in my office, I can look over out of the window here, and most days I'll see a red kite flying past, or several of them, and that's brilliant. And you're probably all aware of the story of the red kite, with the fact that there were just a few breeding pairs left up in the hills near Aberystwyth. But because of a big program to renew or reintroduce the species and stop its hunting and this type of thing, we now got hundreds of them. Oddly enough, I don't know if anybody, maybe somebody in the audience can explain to me later why they don't seem to spread up to North Wales. When I drive up to Carnarvon, which is where my family home is, I seem to see them up to Machynlleth, and I see hardly any of them north of there. I don't know what the reason for that is. However, it's wonderful to have red kites around the place, but on a global level, they're not threatened with extinction to any great extent at all. If you go to Eastern Europe, you'll find very large populations of red kites there. So that's why the IUCN on a global level ranks them as least concern. I'm going to miss that slide. I go to mention again the citrine wax cap, which you may have noticed on the previous slide, is assessed by the IUCN as globally vulnerable. So its threat of extinction is much greater than for the red kite because of the fairly limited populations that you find. It's only found in sort of Western and Central Europe, and the British Isles are a clear stronghold globally for this species. And if you're not sure what vulnerable means, just to point out that the snow leopard and the panda are also assessed as vulnerable. And there's quite a few waxcap species that have this assessment. Some of them, people, when I tell people that, they go, well, that's quite common. And I say, well, it's just like if you're a farmer in certain parts of China, I bet you get quite fed up of giant pandas coming in and wrecking your allotment. So just because they're common here doesn't mean, you need to have a look at the global assessment, and that's the important thing. So we have 15 species of waxcap that you find in Wales that are assessed as vulnerable, and there's even one that's assessed as endangered. But having said that, the amount of funding that goes to the conservation of waxcaps is minimal, and I don't think grasslands get the attention and the protection they deserve in conservation processes, because for all these three species that you can see here, it's habitat loss is the thing that is the threat to them and all the other organisms that share their habitat. So hopefully that's why talks like this, hopefully I'm able to spread the word for people to appreciate how special it's globally important. The populations of waxcaps in Wales are of global importance, and we can't say that for many other organisms at all. So I mentioned the person who christened that term Cheg D fungi was Maurice Rotherow here, who I got to know when I started. I used to know him when I was a student here, and then when I came back to work here, I also got to know about them, and he encouraged me to go and do research on wax caps. Sadly, Maurice died about, well, it was getting on for 20 years ago now, before his time, so I owe him a great debt for introducing me to wax caps. He was introduced to wax caps in turn by Yves Arnolds, who's a very well-known Dutch mycologist. Fortunately, is still alive, and it was Yves Arnolds who coined the term waxcap grasslands from his studies of fungi in fungal habitat, fungi in fungal populations in the Netherlands. The Netherlands is a smaller, more densely populated country than the UK, and he reckoned there was only about 200 hectares. What's that? About less than 500 acres of waxcap grassland remaining in the Netherlands. That's quite a frightening situation. The habitat has all but disappeared there due to intensive agriculture. So the next person who made an important contribution was David Bortman. Some of you may have this book. It's very important to have a good book, a good ID key, not just descriptions. You want nice color pictures, and that's what this book has. He's published a second edition 5 or 10 years ago, which is sadly out of print. I had a copy that he sent me, and I lent it to a student, and I never got it back. So I don't have a copy of this either, which is a bit of a pain. And the last thing we did, which I don't know if it's a service to some people curse us, but me and a load of other mycologists from, I think there's probably about these authors here from about 20 different countries here. We did a global reassessment of the relationships between all the wax caps, which caused us to rename various species into new genera. And I still get people complaining at me for doing this, but we need to put the biology, the fundamental thing in biology is to put things into natural groupings. So that did inevitably require a bit of renaming. So just like Agaricus coccineus became Hygocybe. Fortunately, that one stayed the same, but several of the others changed the genus name. I should mention, if on account of these Bortman books are out of print, this book, I'll mention it later, is wonderful. This is published by, well, it's written and published by David Harris, who lives in Pembroke. It's understated in the title. It says the wax caps of West Wales. You could use it anywhere in Europe and it'd be extremely useful. It's also very cheap. I think if you send David 30 quid, he'll send you 20 copies. So maybe there's 30 people here on the call. If somebody could do that and then share out the copies. So, you know, 3 quid each is so £1.50 each is exceptionally good value. I told him he should charge more. You just have to be careful about the edition. This is his January 2022 edition. So he updates and improves every year. So it's a brilliant resource. Right, back to the story. So David Bortman published this book in 1996, and you can see here, and before then, this is the number of records in Wales for the pink wax cap, which is now called Porpolamopsis calyptriformis. Pink wax cap, everybody knows what you mean. It's the only pink mushroom I've ever seen. And you can see there was, you know, fairly low numbers. I think most of this activity is Morris and various other people early on, but they were spurred on by David Bortman's book and the fact that they could then take the key out and they were singing from the same song sheet, which is important that you're giving things the same name and in a consistent manner. Plant Life helped in 2000 because they did a pink wax cap campaign. And you can see that led to a load more records there. But then after that season, then it kind of fizzled out a bit back to the usual levels. So making people aware of these things is the first part of it. I guess many of you have wandered about and seen a mushroom you have no idea about, and you don't even know how to begin to get an ID for it. So you know, having these guidebooks and having more people familiar with fungi and their ID is a very important thing. It spreads rather slowly, but it does spread effectively. But more broad attention raising or awareness raising is useful. So this is my friend Gary Easton here. We made this pink wax cap hat. So this entertained school children for many years. It's actually very dangerous to wear because it's quite heavy. If you turn your head too quickly, it'll really pull all the muscles in your neck. Reaching a wider audience, this is John Bailey, who I used to know before he retired. He was a really good plant pathologist, but then he changed his interest from small fungi to big fungi. And he and his wife, Doreen, have done a lot of work down around Bristol, the Bristol area. This is Tintsfield Hall. So they got some attention in the national press. So you've got the Times here, possibly slightly less reputably the Daily Mail. But there's a good story. It's nice to see the Daily Mail running this type of story. That's a more positive story than the negative stuff they tend to run, sadly. I've done my bit. I was trying to sort of broaden the issue out and say not just fungi, but if you think about conservation, there's about 80% of conservation effort goes on animals or the attention, and plants maybe get about 15, 16%, and then everything else gets a teeny amount of attention. So I was trying to point out that the great majority of species of genetic diversity are amongst microbes, which would include fungi, bacteria, protozoa, and so forth. So I was trying to argue that we should have a strategy for microbial conservation. So I wrote articles for various journals like New Scientist and this type of thing. I don't think I had much effect, but there you go, had a go. At least I've put it out there and just get a few people thinking about that idea. Anyhow, back to sort of wax caps, the aim of what I was trying to do originally and still trying to do is to sort of prevent the decline in the abundance of wax caps, and that requires certain things. You need a legislative framework. So there are laws that can stop people from destroying wildlife sites. To get the prioritization of these organisms, as a conservation priority, you need to have evidence of decline. I found sometimes that was quite difficult when I was trying to deal with JNCC. They weren't particularly amenable to types of evidence I was trying to find. For bird populations, it's fairly straightforward because there's been detailed observations of birds for at least 50 and often more years, so they can see the declines that have happened. But if there wasn't recording a long time ago, it's more difficult. And for people who are doing actual active conservation, what they want is actual advice. I was talking to Mike earlier about how he should manage his lawn, and I'll mention that in a short while. And then the more pure science part, which I've been sort of fighting with for years because these organisms aren't easy to work with, is the understanding the ecology, the ecological niche and the habitat requirements of these grassland fungi. And lastly, which turned out to be easier and has been more useful, has been sort of finding better ways of doing distribution, assessments of distribution. Usually mycologists would do a survey of fruit bodies, but to do that, you need to be a very confident and experienced identifier. So you have people up in, I'm sure many of you would know Debbie Evans and Charles Adam. They're the sort of North Wales experts, but they're really busy. I think Debbie was telling me she'd been sort of full speed at Shirk Castle, I think for quite a few days. Because the mushroom season is so short, people like Debbie and Charles were in very short supply. So I was trying to devise a method that would be slightly easier in that you didn't have to look for the fruit bodies, you could detect the DNA of these fungi in the soil. So I'll hopefully get onto that. I'm going to jump a little bit because Mike said, when he heard me speak earlier, I ad-libbed so much on the earlier part, I didn't get to the end part. So I'm just going to very quickly go through the legislative stuff. Llanellishen Reservoir in Cardiff is an SSSI because of its waxcap populations. And closer to you at the moment is a Thynog in Bangor, Brewery Fields. That was notified as an SSSI based on its grassland fungal, or in parts based on its grassland fungal populations. And so the council, the Gwynedd Council, wanted to build a Ysgol High Top, I think, on a large part of it. I think it was quite a good results because they ended up with classrooms and mushrooms by reaching a compromise, and that's usually how these disputes are usually resolved. I think I will jump this part. This is evidence decline. Only to point out that, I'll have to say something about this. Park grass is the world's oldest experiment in Rothamsted near Harpenden. And back in the middle of the 1800s, they set up a load of of agricultural treatments. These would be, that one there is farmyard manure, and in different levels of lime going from right to left and so forth. And what we found, that's a very simplified design of the whole thing, of that field. But what we found when we've gone and looked for wax caps on park grass is that they're virtually all in the areas which have had nothing. So that plot there that I've got the arrow there has had nothing at all added to it for 150 years. That one's had a teeny bit of lime added. This one's had nothing at all, and that's had a teeny bit of lime. All the plots with nothing on them at all have all had a lot of nitrogen applied. And you can see there's a few things where there's phosphorus applied. So farmers usually apply NPK fertilizers. So of the components of that, it's the N that seems to be the most damaging to the waxcap populations. And had they not done that, you can see this is quite a good waxcap site there, all the species we found. I think when we did the DNA stuff, we found a few more as well. Right, so that's evidence of decline. I skipped over that rather quickly. Basically, there would have been wax caps over all that field, and the wax caps are left only in the place where where nothing was added. So modern agriculture, even without ploughing, can be quite damaging for wax caps. So the Wildlife Trust all the Wildlife Trusts have lots of grassland field sites, and I often get asked by the people who have to manage them, we've got some wax caps there, how do we help them along? How do we promote their fruiting? And because lots of important wax cap sites are lawns by country houses, so they're ancient grasslands that have been mown continually, it gives the impression that you need to mow your grassland all year round, all the summer, which is, well, apart from being rather a lot of work, it also means that you get all your wildflowers and the associated invertebrate populations are going to be diminished. And so people often thought they had to choose either between having wax caps or having the wild plants. So we explored this in a bit more detail. So this is part of my job as a scientist, is that rather than, often you hear anecdotes about what's good, and those anecdotes are probably correct, but I wanted to provide some robust scientific proof for this. So we did an experiment in Bala, in Llanuwchllyn. There's a farm called Trawscoed that is owned by Andrew Graham. Some of you may know Andrew. He's a very accomplished entomologist as well as being a mycologist. Anyway, he was kind enough to let me use a field that he had. You can see that in the bottom right here, and I split it up into lots of little plots like a checkerboard, and we applied a load of different treatments. So in one plot here, 3 cm in the graph, we use this ride-on mower, and we cut the grass every 2 weeks to 3 centimeters. That's like a lawn. That's how a lawn would be managed. On another set of plots, we didn't do any cutting until late July. And then we used a scythe to cut the main bulk of grass, like a hay cut. And then after the hay was removed, we then cut the grass to 3 centimeters till the end of September. So that's the traditional hay cut regime. I'm sure the Wildlife Trusts use that. That's a sort of standard thing. The only difference is, I think, that it's quite difficult to arrange the aftermath grazing or the mowing. So often you see these wildlife sites and there's been a lot of regrowth of the vegetation after the hay cut, and the wax caps don't like that. They don't like it to make mushrooms. I'll explain that in a second. Third treatment here was a very late hay cut. And then we had 2 treatments here, which is the same as the first 2, but we only cut to 8 centimeters. And then the last one was not cutting at all, and we found hardly any mushrooms at all on that. So the good news here is that you get lots of mushrooms if you do a hay cut, as long as you keep the sward short in August and September. And the reason for that is that mushrooms, like I said earlier, that the only purpose of a mushroom is to spread the spores. And if the grass is long, the spore spreading business, which relies on wind blowing across the mushroom, will be very inefficient. So the fungi realize this. I think they probably sense the carbon dioxide levels at ground level, and they don't bother making the mushrooms if the grass is too long. So if the grass is very short, then they produce more mushrooms. Having said that, the mycelium underneath the ground is probably perfectly happy. So you're not killing them off by not cutting the grass. And even in these uncut plots, the mycelium, which is 99% of the biomass of the fungus, is perfectly happy and unaffected. So if you don't cut the grass for a couple of years, that's probably fine. You just have to be careful that if you keep on leaving the grass uncut, then eventually you're going to have successional processes happening and trees will grow. All right, so now this is the stuff I try and do as part of the more detailed research to understand the ecology and the nutritional requirements of these fungi. The first thing I found fairly quickly is that wax caps are not model organisms. An ideal model organism is one I could catch in the wild, if you like, collect, bring it to the lab and grow it in the lab, or do stuff with it in the lab. Very difficult to do for wax caps. If I try and grow them, their spores hardly ever germinate. This is on agar plates, little petri dishes. I tried everything. We tried for years and we failed. This is the best we got. They do this. There's the spore. It's produced a germ tube in this strange structure. It does nothing else. The snowy wax cap, there's the spore, produces a little germ tube there and stops. Conica, Hagocybe conica, the blackening wax cap, doesn't even produce a germ tube. It looks like it's trying to, but it doesn't. and the parrot wax cap produces a germ tube and then this strange structure here. Most of the other species, especially the rarer ones, didn't do anything at all. We tried growing it from the fruit body, from the mushroom, tissues of the mushroom, that didn't work either. So that means we can't do any growing of the fungus, because if we could do that, we could see how does it like nitrate. Does it like nitrate or ammonia nitrogen or protein nitrogen? We could have done those experiments, but we can't do them. So we have to look elsewhere. So one part of the elsewhere was looking at the phylogeny, the related, within the family Hygropheraceae, which is where all the wax caps are. Do we find any useful information from the other things in that family that aren't wax caps or don't get called wax caps? And we do, because there's one genus, Hygrophorus, and that doesn't grow in grasslands, that grows in woodlands, associated usually with conifer trees, and it forms a mycorrhizal association, an ectomycorrhizal association with those trees, just like chanterelle and porcini, and most of the woodland mushrooms you would find. They're mostly ectomycorrhizal, and they rely, those species, ectomycorrhizal fungi, they associate with the roots of the trees, and they provide nitrogen and phosphorus to the trees in exchange for sugar. That's how they earn their crust. Another group within the family Hygropheraceae, so all these ones at the top, they're wax caps. There's more wax caps, there's more wax caps, wax caps, wax caps, wax caps there. So you can see most, I'm pointing out the exceptions that prove the rule here. So you've got the ectomycorrhizal group genus here, Hygrophorus. You also have some that are lichenized. So here you see, here's a lichen here, and in the autumn, this lichen produces a mushroom. It's called a basidiolichen. The vast majority of lichens are cup fungi, but there are a few groups of basidiomycetes that are lichenized like this. So we find this if you go to the tops of mountains in Snowdonia, sometimes you'll find this yellow lichen Omphalia. It looks very much like a wax cap, but if you look around the base of it, you'll see there's this green crust. That's the photobiont. That's the algae that it associates with. And if you go to Brazil, so tropical areas, you find Dictynema and Cora, which basically you have a lichen on tree bark, and every year it makes mushrooms from there. So based on these two oddities within the family, it suggests that this whole group of fungi maybe are biotrophic. That means they associate with a plant and they get some nutrition from the plant, just like the Hygrophorus and the lichen Omphalia. So I've been trying to provide evidence or discover evidence that says that that's what the wax caps are also doing, but it's quite a hard job, as I'll show you. So one thing you can do, you probably have heard about archaeologists. When they find a human body, they can, they'll do all sorts of. Archaeology has been transformed by scientific advances, so they can get DNA out of the dentine of the teeth, and you can sequence the whole genome. That's what Svante Paavo got the Nobel Prize for last week, that you may have noticed in the news. But as well as the DNA, you can also study the isotopes that you get in the teeth, and that can tell you stuff about where the person was born, what their diet was, and so forth. Now, the isotopes they look at aren't the radioactive isotopes that people often think of. They are mostly the stable isotopes. So basically, it's an element with an extra neutron in the atomic nucleus, which might make sense to some people. So, for example, the molecular weight of carbon is usually 12, but if you have an extra neutron in the atomic nucleus, then the molecular weight has gone up to 13. So you can measure the relative amounts of the 12 carbon and the 13 carbon with something called a mass spectrometer. And similarly for nitrogen here. So here's an example in this graph of a marine food chain. So you've got the particulate, that's just particulate organic matter is just dead. That's just basically dead stuff. It's like the organic matter in soil. And then you have the primary producers, the seaweed and the algae and the other, or the diatoms and so forth. And then they're consumed by amphipods, bivalves, filter feeders, and then they're eaten by walruses, various fish, marine birds, and then all of them are eaten by seals, and seals get eaten by polar bears. So you have like a food chain here, and you can see as you go up the food chain, the relative abundance of the heavy isotope of nitrogen increases in the body of the polar bear relative to the body of the kelp or the algae here, and also the relative amount of the heavy carbon, the 13 carbon, also increases here. Now, these numbers are all negative because it's don't worry, that's, you can see they're becoming less negative, so they're increasing. So you could do the same thing with people. I'm sure there's a couple of vegetarians in the audience. If we took your hair and did isotope analysis, the vegetarians would have 15N levels about here, about 10 per mil, whereas the ardent carnivores will be up here about 20 per mil. You can also do things, you can, if you took the hair from a person who's been living in America, you'd find it was down, it was here, because they eat mostly maize, whereas we eat mostly wheat, and we would be down here. So that's how you are what you eat if you're a human, if you're in the marine food chain, and also if you're a mushroom. So we could take a mushroom and have a look at the isotopes. And when I first did this, it was quite perplexing because they were really strange. I did them again because I thought the machine was broken. They provide a distinct group here. I've done lots of these, eventually published a paper after umming and ahhing about this for a long time. They are quite distinct. They're very enriched in the heavy nitrogen isotope and very depleted in the carbon isotope. If you put higher plants here, they would be about here, about -30. So it seems to be that's kind of suggestive of the fact that they're getting sugar from plants. If we look at ectomycorrhizas in woodlands, we find them to be a little bit enriched in nitrogen, more so than the non-mycorrhizal, the saprotrophic fungi, the fungi that are either on the soil, growing on soil organic matter, or growing on wood. We find them reduced here. So if you take a trajectory from the saprotrophs here up to ectomycorrhizals, it looks like the wax caps are supramycorrhizal in some way. We're not really quite sure why. They are very enriched in this heavy nitrogen. So that suggests in part that they're giving the lighter nitrogen to the plants in exchange for the carbon, but there's more to the story than that. I think they're getting the nitrogen they're obtaining is from an enriched source in the first place. And hopefully I didn't make that too confusing. Another point, I think I heard somebody talk on the chat earlier. They were saying that they found a wax cap in a beech woodland. That's quite unusual. Beech trees are ectomycorrhizal, and I think what happens there is that wax caps get beaten up by the ectomycorrhizal fungi, these organisms, and that's why you don't find them very often. There's an example of one I found in the Burren, the blackening wax cap in a hazel woodland here. However, if you go, that's our woodlands, you don't find any wax caps, but if you go to other parts of the world, you can see these X's here. This is where the samples for this graph here came from all over the world. If you go to New Zealand, New Zealand's been quite badly trashed by Europeans since they got there, but their natural forest type is a laurel forest. And similarly, if you go to Tenerife, if you go to the mountains in Tenerife, you'll find they've got lots of laurel forests. And if you go in the autumn, you'll find loads of wax caps there. And if you go to the Amazon rainforest and the forest of Central Africa, there are lots of non-ectomycorrhizal trees, and you'll find quite commonly wax caps in dark bits of rainforest. So I think it's quite intriguing. So it's a European perception that wax caps are mostly found in grasslands. And I think they would like to live in the woodlands, but they get beaten up by the ectomycorrhizals, because the ectomycorrhizal fungi can't grow in the grassland because their tree host isn't there. So on the right, I'm sort of summarizing here the evidence I've got, which still hasn't convinced quite a few of my mycologist colleagues, but it's difficult. And to get to do more on this, you have to get funding, and because this isn't prioritized, it's rather difficult to do that. And these would be you'd have to do some quite big experiments to get more robust evidence. So in terms of their nutrients, that's the nitrogen and so forth. They have the wax cap and the other chaga fungi have very distinctive 15 nitrogen patterns, which suggests that whatever they're doing it's probably mycorrhizal, but they're all doing the same thing. So the fairy clubs and the earth tongues all have that weird pattern of the 15N13C. About 20 years ago, I was involved in some work in, what's it called, the NERC Soil Biodiversity Programme, and there was a grassland in Scotland where they imposed various treatments, and I was looking at the wax caps there, and I'm still perplexed by the fact that on one plot where they applied lots of Dursban, which is an organic phosphate insecticide, that had a clear negative effect on the number of wax caps. There are no wax caps. If you kill all the soil animals, you don't get any wax caps forming. Possibly, the soil animals have quite a high level of the 15N, and maybe that's what the wax caps do, is they feed on the bodies of the soil animals. I think I did some sums to sort of suggest that was possible, but the evidence is still a little bit circumstantial. In terms of the energy, where they get their energy from, the sugars, we use there's a radioactive carbon 14C. So we didn't add this, that's naturally present at very low levels. And it's taken up from 14C labeled carbon dioxide, and we found that it gets into the mushrooms fairly quickly, which suggests that the mushrooms are getting their nutrition from living plants. However, if you look in a grassland, you see a whole range of plants, and you're trying to work out which of those plants are the wax caps associating with. I think they're probably associating with nearly all of them. People used to think that wax caps associated with mosses, because you very often find wax caps in mossy grasslands, but mosses don't really have roots, so there's nothing to associate with. And if you go to places like park grass, there's hardly any moss there because it's a much drier climate. So you don't always have to you see wax caps where there aren't mosses. So I think they're probably quite Catholic in their taste of host. They'll be associated with dandelions, hawkweeds, grasses, legumes. That's what I think is likely. We've had a look at the roots of plants, and you can see here, here's the root of a plant that was from near where there was a mushroom growing. We had a look at all the fine roots, and you can see there's a little hypha, a fungal mycelium growing through there. And we know it's the mycelium of a Basidiomycete because it's got a thing called a clamp connection there. You can see it there as, again, the little bump. Only mushroom-forming fungi produce them. And also, we could get DNA out of these roots and do a specific test, for a PCR test. Everybody knows about these nowadays because of the COVID test. Remember the PCR test that people used to have before they had the lateral flows? Same idea. So we're detecting the DNA of the fungus inside the roots, just like they were detecting the DNA, the RNA of the virus inside your sputum. So we see that as well. What we can't see is an obvious mechanism for the exchange of the nutrients, and that's where my picky mycologist colleagues always shoot me down. But that's the idea. That's what they're supposed to do. You know, that's the whole point. If you're a scientist, you're supposed to be cynical and try and knock your own idea down. So I haven't quite got there with that, but I'm working on it. So the last part was the DNA sequencing, the eDNA. The eDNA stands for environmental DNA. So what we do here is, this is Andrew Graham's farm near Bala, and it's a field next door to the one we did the mowing experiment in. And I've laid down a tape here. There's a tape across there. There's a 30 by 30 quadrat, and we take a soil corer, a soil auger like this. This is when we started. We didn't have any augers, and that's actually an apple corer. That got quite painful doing that work. But we'd go along every 5 meters. We'd take a core, so we'd end up with 36 cores from that quadrat, which we put in the same bag. We took it back to Aberystwyth. We froze it, and then we did some freeze drying on it, which takes all the liquid out of it, whilst it's still actually, with no water being present, at low vacuum pressure. So basically dries it out without allowing opportunity for the material to degrade. Then we grind it, and then we grind it even finer, and then we take a small amount of the dust, about 1/5 of a gram of the dust. That's about 1/2 kilo. So we're subsampling and then subsampling again. We take the DNA and we do more PCR on it, similar to the one this PCR I was talking about here. But here we PCR all the DNA from all the fungi for the particular barcode locus that we use. And then we sequence all that DNA. We can only do that because we have devices now that these devices like this Iron Torrent personal genome machine weren't made or designed for people like me to do ecology experiments. They were designed to sequence human genomes to look for genetic defects that cause cancer. So one of the things they'd like to do, I don't know if any of you are aware of it, but if you have a developing cancer in your body, way before you feel any symptoms or it can obviously be detected, there are just a few cells from the tumor floating about in your blood. So if you can take a blood sample and sequence, I don't know what the technical term is, sequence the crap out of the, sequence it very deeply, you do lots of sequencing of it. If you can detect genetic signals of the tumor, then you then have a diagnosis, and then when you apply a treatment, an anti-cancer treatment, that amount of DNA should go down, in which case you know you're winning the battle. So that's why they have these devices, but we can use them very effectively for asking questions of soil biodiversity. It tells you what you end up with after you've done all this sequencing is a list of all the fungi there, and also which ones are more abundant and which ones are less abundant. So you get the relative proportions. So before I show you any data of that, I'll just show you a site that we quite often use. It's a bit like park grass. This is the Aberystwyth version of park grass. It's up in the hills by Devil's Bridge. And we've got various treatments that a hill farmer in Wales would use. We've got hay cut and aftermath grazing. We've got summer grazing, which is the more common thing, and the addition of lime or not. And here's the list of species we get. This is these are only the more common species from DNA sequencing. So they're the species there, and you can see the ones I've highlighted are the wax caps, the earth tongues, the fairy clubs, and so forth. So they're very common in the soil, which is kind of as expected because it's a relatively undisturbed upland grassland. However, the only treatment where there's not quite large numbers of these things is this one. This has got some other fungi in here. These top three aren't wax caps or chegg fungi. And if you look at the amounts of the chegg fungi here, there's hardly anything there at all. That's because this is the only plot that received the fertilizer, the chemical fertilizer. Yeah, the other ones are different, but the other ones we find some differences with, but the most different one is the one they add the fertilizer to. Okay, so that's kind of illustrating that even on these ancient grasslands, you can reduce the abundance of the wax caps, even if you didn't go and plough. Possibly ploughing is more damaging than fertilizing, but both are best avoided if you want to have good wax cap populations. Here, this is the work we've done before we did the DNA sequencing. This shows that on the fertilized plot, we didn't find any mushrooms, and we know we didn't find any mushrooms because there's none of the mushrooms there. There's no mycelium in the soil. For some of the other plots, you can see there's two plots here where we find quite a few mushrooms, and these ones we didn't find many, and that's because they were hay cut, but not aftermath grazed. And therefore, if there's long grass in the autumn, as I mentioned earlier, you don't find much fruiting. But the wax caps are there in the soil, and they're quite happy. I'm just going to skip that, and I'll go to this one. So we've got this method we developed, and we want to find a use for it, if you like, or make it useful in fungal conservation. So the opportunity came up in 2015 when this guy here I'm pointing to now, Anthony Ravenscroft, he was a student here in Aberystwyth, and he did countryside management, and he'd heard of the stuff that I was doing with the eDNA. So him and his pal Perry here, they work for Dudley Council, and they look after this site in Hales Owen. So it's suburban Birmingham. It's not a very promising area. You wouldn't think to go looking for wildlife sites in the suburbs of Birmingham. But this place, the Lisos Country Park, you can see a map here. You can see it's surrounded by housing. The M6 is only about a mile away. The cradle of the whole global industrial revolution is about 5 miles away from here. So it's quite surprising that you have any wildlife of note left behind, but this is quite steep. You can see these quite steep slopes here. So I guess when they're developing the area, they decided to not build here, they'd build around it. So they have this country park, and with the help of this guy here, Nick Williams, who was a lecturer at one of the local colleges, he had been looking at the wax caps since 1997. I point out there that in 1997, I don't think I knew what a wax cap was. Anyway, he had these counts, and over almost 20 years, he'd found 22 species around here. So this is obviously quite a special site. So they asked me to come and do some soil sampling and eDNA, and we sampled these green quadrats here from the areas, the best areas. He had names like Scarlet. There's one's called Punicea Ridge, I think is one of them. They've got names that relate to the fungi, so they were the good areas. So we did the soil sampling like I described there, and this is the species list we found. So they're the 15 areas we sampled here, and these are the fairy clubs at the top. These are the earth tongues, and these are the wax caps. So you can see that we didn't find five of the species that Nick had found before in yellow here. We didn't find, but we did find, I think it was 6 species that he had missed. So the good thing about that is he'd had 22 species, and we could potentially add 6 to that list. And 28 species is an impressive amount. That's what there are at Llanishen Reservoir, and that's an SSSI. And they wanted to get this site notified as an SSSI. However, at the moment, the rules for notifying SSSIs, they talk about mature individuals. So basically, if you've got a mature individual is like a mushroom. By definition, if you can see the mushroom, that's a mature individual because it's reproducing sexually and producing spores. But if I detect mycelium in the soil, I can't guarantee that is a mature individual. So these 6 extra species that we found couldn't be counted unless we could actually find the fruit bodies. So, but one thing I could do helpfully was say, I could tell them exactly where the DNA of the species they'd missed were. So Hygocybe conica, I hadn't found that, and that was in this little square here. Intermedia was here, and Spadicea was here. So we could go, I told them the following autumn, I think went February 15. So in autumn 2015, I said, go and have a look for these species. And they found those 3. And they also found this one. This is not a wax cap, it's a green earth tongue. I haven't mentioned these so far. They're distantly related to those black earth tongues. And that was found, I told them it would be there, and I said, go look for a very small green thing, and they found that as well. Okay, so we added, of the 6 species of wax cap that we said were there, they found 4 of them, and they also found this one, which we also predicted. I call this eDNA guided surveying. The reason I'm drawing attention to this one is that this is one of those species that's very rare and punches above its weight. It's in England, they call it section 41. In Wales, we have section 7 of the NERC Act. So that's a species that punches above its weight. So it's more helpful to, if you're trying to get legal protection. So there's an awful lot of paperwork with making an SSSI, and it took another couple of years up to 2019 for the SSSI status to be confirmed there. I think we also found we had to apart from that's the area of the country park, we focused our initial attention there, but then we did some more surveying around here on this golf course, and we found a couple more species as well. So that's the first time globally that anybody's managed to use eDNA evidence to achieve legal protection for a site. To do this research, I have to get funds in to employ Andrew, who's my colleague. I think there was a picture of him earlier, to do the lab work. And then we jointly rewrite the reports. Here's one for Butlins, or Haven Holidays as they're called in Morfa Bychan, and they wanted to put caravans on here. So Gwynedd Council asked me to do an eDNA survey there. We found some wax caps, but nothing special. So unfortunately, we couldn't say this is we couldn't find a compelling reason to prevent development. So that's like a sad news. They haven't put caravans on there for some. I went past there recently. This is after 8 or 9 years, and there's still no caravans. So that's quite good. The other thing we do is for the Welsh government. If you own a farm and you have a a pasture, an old pasture, and you want to plough it or plant trees on it, if you want to change its use, you have to get an EIA screen, an environmental impact assessment screen. And I always notice these Hymax. You probably know. I'm sure many of you notice these little diggers in small fields that are working at weekends, and you wonder how does that landowner have permission for doing that scrubbing out of a hedge or whatever it is, and probably not. That's what they're doing at weekends because they're less likely to get caught. Maybe, I'm not sure. Anyhow, if the EIA unit of the Welsh government find out a farmer's done that, they can fine them quite heavily and enforce some restitution as well. And they asked me if we'd see if eDNA would be helpful for them in proving the guilt of a farmer. So we did this site down in Pembrokeshire, I think it was, where they had this nice heathland here, and then suddenly there was a wheat field where there used to be more heathland, and we could detect various fungi and also plants that were there in the eDNA in the soil that demonstrated that wasn't always a wheat field as the farmer had claimed. I've mentioned David and the Pembrokeshire Fungus Recording Network. That's David Harris. And these guides he's got. So it doesn't just have the wax cap one. That's the older version compared to this one that I'm showing now. This is the later edition. He's got a sand dune one as well. They're very good and very cheap. So he's doing them at cost essentially. And David's also doing DNA barcoding. He's set up his garage. It doesn't look like a garage to me. He calls it a garage, but it looks more like a kitchen to me, or a back room. He can do DNA barcoding. So you can collect mushrooms that look a bit unusual, and he gets a DNA barcode. All that stuff we do with the DNA sequencing stuff relies on having, I have to be able to link the sequence to a mushroom which has a known name. And part of this work that he's done, he found, and we're just writing a paper for a new species of wax cap that we found down in South Wales that's new to science, has never been found before. So citizen scientists are very useful in doing all this type of work. So if any people up around Conwy are keen on this, I can introduce you to Brian Douglas, who was one of my students, PhD students. He works at Kew now, and he's very keen on spreading this type of technology around to citizen scientists. I'll stop there with a thank you for all the people who've helped. I've probably missed lots of people off here, but there you go. Thank you very much for your attention. I hope I didn't send you all to sleep.
I don't think so. I think I've been concentrating really hard, actually, extraordinarily hard to try and take it all in. What a fantastic presentation! Thank you very much. Yeah, I might have to see that yet again to kind of take it all in, to be honest, Gareth. It's the third time watching.
He's got it recorded now, so hopefully I didn't slander anybody.
Not at all. While people might be thinking of questions, I mean, your appeal for citizen scientists at the end was quite interesting, really, because I'm sure there are people within the wildlife trust, really, particularly in the Conwy Valley, who might have plots of their own, which they might want to either change the management of, or maybe how much the eDNA. Is that something you can buy, or just?
It's only us that do it here, really. Yeah. Other people could do it, but they don't. I suppose that you need to have the knowledge. The sequencing part is fairly easy. It's the knowledge. It's the taxonomic knowledge that's more difficult or more limited. Yeah, we do. But that's how I keep Andrew employed. Is Natural England, Natural Resources Wales, National Trust, to people like that wanting samples. About 300 quid for a sample.
All right. Yeah. Right. Okay.
I think I keep it. That's actually not too bad, because by the time you've paid, if you can get hold of a very experienced mycologist like Debbie, she's so busy. Debbie, I think most of you know Debbie Evans, or many of you would know.
Yeah, we do. Yeah, absolutely.
Yeah. So there's not many Debbies, so it's a matter of if you could get her. I think Debbie does. I keep on telling Debbie she should rate her prices, but she doesn't do it for the money. She does it for the love of the work. Right. But I think a better way of doing it is if people find a site they think might be good, you do eDNA first, and then you have a list. You have a species list of things that should be there. And you hand it over to somebody like Debbie, who is quicker for her to then go. She can go. She can visit the better sites. Right. Focus their effort. Because I think the cost of a survey, it would probably take a couple of days to do a survey and write it up, and that might be 300 quid anyway.
Okay. Okay.
I think I think the economics of it is level pegging, I'd say.
Okay. Is that the kind of thing she's doing in Chirk Castle now? Then is that sort of?
Yeah. Well, I'd done some eDNA stuff there before for another reason, so I did tell her there was one of those rare green earth tongues in a particular place, so she's probably having a good look for that at the moment. The other thing often with those planning requests, if it's a planning thing, they're always in a hurry. Planners are always wanting the results tomorrow.
Yes.
They're asking you in January, and you're telling them, well, you've actually got to wait till November before we can tell you. They get quite annoyed about that. But with this method, it doesn't matter. You can do it in February. You can do it any time of year, and it'll give you the same result.
That's right. That's right.
Because the mycelium is there, all year round. It's there for, they're probably longer lived organisms than we are.
Right. Okay. So that the finding the presence of mycelium, can that stop the planning application? Is that can they actually do that?
Yeah, I don't. Well, I think it definitely is there. It's not necessarily a mature individual.
Yeah,
that's the grey area. But then I think I don't know if NRW were given the evidence that the DNA was there, they would say, well, you've got to wait till we get Debbie or somebody to go and have a look and see if she can find the mushrooms.
Okay.
We're doing something like that in Llanberis at the moment.
Okay.
And I didn't know. There was an even rarer fungus there called Porpulomopsis, or pseudotrichoma. It's called. It's globally endangered, so it's a particularly rare thing. I found it, and I was quite surprised because I hardly ever see it in the soil eDNA. Debbie also found it.
Okay.
So she knew in the same place. So that's good evidence. That's good evidence to protect that particular set of fields.
Okay. Is there kind of a time when a fungus becomes mature? Is it kind of there's a time scale that you know about? Is it, I don't know, 10?
No, we don't know. Yeah.
Yeah.
But, some of you might know Sam Bosanquet. He's the specialist at NRW on mosses and what they call annoyingly they call lower plants and fungi. Anyway, Sam knows of a lawn near where he lives, where half the lawn was ploughed up in World War II for potatoes and the other half wasn't. And there's tons of wax caps on the unploughed bit. And we did the eDNA on the both bits as well. And he's done the surveys. I didn't compare notes with him until this morning. And we find exactly the same thing. But something that happens, World War II was when? Getting on for 75 years ago.
That's right, yeah. More
than 75 years ago. And there's still, the ploughed bit is still rubbish for wax caps. There's some wax caps there, but compared to the other bit, it's rubbish. And I think he also said there was succisa was in one part. That succisa wasn't in the ploughed bit either, even though it's right next door. So the inoculum and the seeds and everything could blow over very easily. The damage is lasting almost a century.
Okay, I'm not taking over all the questions. Does anybody else want to ask a question? If you want to put your hand up or just unmute yourself and show yourself, by all means do so. There's a question in the chat from Julie. I don't know if you want to ask your question yourself, Julie, but I can say it for you if you want. Can you read it? Yeah.
Yeah, you can read it. Okay, great, thank you.
Can I read it? Okay. He said,
he's reading it. So.
Okay, I'll read it now so we all know what it is. Keep Wellstide are given a grant to a churchyard for a community garden, including returfing for wildflowers. She's found an amethyst, coral, nitrous, one earth tongue, meadow, and a couple of reds and yellows. Should this need an EIA? Yeah.
Returfing is then adding turf on top of it, is it?
They're going to completely, they're going to scarify it. They're going to lift the old turf, and they've got some turf available for wildflowers, which has got wildflowers growing in them. So they're trying to do a bit of rewilding, which is nice, but obviously it will totally destroy any mycelium.
Yeah. That's what seems a bit silly. Why don't they just? What they could do, you always end up with compromises here. You could, if they've got this turf, I didn't know wildflower turf was a thing.
Yes, and nor did I until today.
Where did it come from? Presumably somebody else is losing their wildflowers as well, are they?
It's a bit of a worry. It could be from South Wales. It's definitely Wales, but it's not necessarily local to here.
No.
We're up in North Wales.
Yeah. Maybe what they could do as a compromise is just do a few squares with the new turf and leave the rest of it alone.
Unfortunately, it's quite a small area, and they've given them grants for a shed, a greenhouse, 3 big raised beds. I'm just kind of wondering really whether that kind of level of wax cap is an interesting site or not really.
Well, nitrous wax, what's that, neohygrocybium nitratus?
Yeah.
I think it's globally vulnerable.
Really? Okay, well, I might be wrong. It was kind of one of these lilac-y, and everyone seemed to feel that it was quite smelly.
Lilac? No.
Yeah, yes, it was slightly lilac-y brown.
I did a lecture this morning to undergraduate students, and I was telling them off. I said, I bet none of you like Latin names. And they said, well, tough shit. This is what you have to do if you want to be a scientist. You have to get familiar with them. So I kind of practice what I preach, and I'm really bad at recognizing the common names for fungi. I just realized amethyst coral is Calaveria zolingeri, isn't it?
John Ratcliffe might know the answer to that one. Can't remember.
It's violet in color.
Yeah, violet in color. Yeah.
A big.
Yeah, it's a big patch.
That's definitely vulnerable. That's really special.
Right.
They definitely that's way more important in terms of preserving rare threatened biodiversity. Those 2 species that you mentioned there are way more important. And you say there's reds there. That sounds like a pretty good site. They shouldn't go messing about with it.
Right.
Agreed.
Yeah. So.
Maybe they're doing it. They're doing it with the best will in the world. They're just.
I know.
It's a bit of lack of education on these issues, isn't it?
Yeah.
Chris wins something.
It's a really difficult situation because there's a couple of friends involved with it, but I might have to. It is quite a small area. Someone said something about 0.2 of a hectare. It's probably less than that.
They're still quite special. If they could get all the other plants, they don't need to introduce plants from elsewhere. If they're clearly managing it correctly. I mean, I'm just wondering why the diversity of plants isn't there already. Maybe it's just been cut very short for a long time. Yes,
probably. But the trouble is, they're deep raised beds, and everything else will trash the rest of it.
Yeah.
They could put that in though. They don't need to put it there.
Yeah, I think Chris Windsor put something in the chat as a reply there. He says, Julie, I think there's a minimum size at which the EIA will be applied, about 0.2 hectare. But it does sound like someone needs to talk to them, he thinks.
They sound like nice people. They're just, the worst you could say about them is they're misguided, isn't it?
Oh yeah, absolutely. They're delightful. I don't want to upset them, but I think I'm going to.
You just have to say, we didn't really. I just realised these things were really rare.
Yeah.
If you want to find out the rare fungi, I'm just looking on the Global Fungal Red List Initiative, because it's a bit of a bureaucratic process for a fungus to get the global red list assessment, and there's quite a few species in the pipeline, if you like. I'm pretty sure.
Could you maybe put that in the chat?
Yeah, sorry, I'm looking on my.
Chris Wynn's got his hand up.
Do you want to join it, Chris? You've got your hand up. Do you want to join in?
Yeah. Now, all I was going to say, if Julia gets in touch with us and gives us a few more details, perhaps we can talk to either Keep Wales Tidy or the community group, and just give them a little bit more advice and to outline, the interest really, and see what happens from there. Yeah, so just drop me an email or give us a ring at the office. See what we can do.
Can you put your email into the chat for us, Chris?
Yeah, do that.
You can pick it up. Yeah. If people don't know who Chris Wynn is, what's your title, Chris?
Senior Reserve Manager.
Absolutely.
So, I like to stick my oar into other things as well.
Well, please, yeah. I'm glad you come along. It's great to see you.
No, it's really fascinating.
I know it's take me back to my
great science.
It's science, isn't it? Take me back to my undergrad.
Yeah, it's really good.
Just trying to figure out graphs and kind of stuff. It's been mesmerising, really, and it's so interesting. And I don't think many people do realise how I mean, the waxcap look great, don't they? They look very colourful. They look fantastic. We want to see them, but they're important, aren't they? And they're important for species which Wales has a lot of, and I think we should kind of protect them much more than perhaps we are doing. Yeah, I think there was an event there today, wasn't there, at Athinog? Did anybody go to that to see the wax caps? Was it a kind of an event? I wonder if anybody might have kind of tried to go, but it was booked up. I think that was this morning, wasn't it? Did anybody go? Perhaps not.
Unfortunately, I couldn't go.
Athinog, the person who deserves the credit there, I think, is Muredydd, isn't it?
Yeah,
He did all that very painstaking searching and found species that were very hard to find.
He did, yeah, and he's been involved for decades now since the site was first protected, and working with Charles as well.
Yeah,
we've had various surveys done looking at the fungi ever since, and it seems to be coming along really well. At Athinog, we've been using cattle grazing to help us get the sort of short sward at the right time of the year that you were talking about, Gareth.
all right, yeah,
and that seems to be really working. And doing really well. So we're finding new species almost every time we do a survey. So yeah, it's great.
It's the one I used to go to. My dad used to take me to it a lot. Was Tanabulch up in Clonnog.
Oh yeah.
That must be quite difficult getting the grass was always long in the autumn, which I imagine was because it's quite difficult to get the grazing animals on there.
Yeah, I mean, Tanabulch it's always been a difficult one. I mean, at the moment, it's doing really, really well for the right butterfly orchids. In the last couple of years, we've had astronomical counts. It's been great. But one of the difficulties has always been getting the aftermath grazing right, because it's so wet. I can't remember for how many years now, but we've been using sheep rather than cattle to do the aftermath grazing, because it was the cattle who were tending to poach the ground, and that was making it sort of a bit rank. So, but we've been using sheep now, and they seem to be working better. But I'm not sure we've ever done anything really detailed by way of looking at the fungi there, and that would be quite interesting. Although
past there, because it's right, the hills around there are really good for wax caps generally.
Right. Okay.
And I always stop off and have a look. So if I see the grass long, I'm just going to get wet trousers and I know wax caps, so I don't go in.
Right. Something to look at for the future, I think. Yeah.
I know it's hard. It's hard. The grazing animal issue is quite hard, isn't it, with all the regulations?
Yeah, it is. Yeah, and the tricky thing is he's getting it right, isn't it? He's getting the sort of, like you say, getting the right conditions for the fungi at just the right time. And each year it can vary subtly, can't it, depending on what the weather's been like. And it'd be interesting to see this year how well the fungi do at Athinog, given that it was so dry for quite a long period.
Yeah, I hope you get success with this small site with the church people, because that seems it's like a scope for education there, isn't there?
Yeah, indeed, yeah.
Keep Wales tidy as well. The funders.
I'll put my email address in the chat, and Julie just drops me a line.
Okay, that's great, Chris. Thank you very much. Thank you. Smash it. Thank you.
If you want to find out which the rarest wax caps are, or fungi generally, I put the Red List Initiative website in there. So you just type in, you search the species and start typing its name, and the various species pop up. So that the amethyst coral is definitely in there. Nitrous wax cap is definitely there. I wouldn't be surprised if some of the reds are also on there. So that's a panda level rarity. If you tell them that, as rare as a panda.
Okay.
At least they've got an animal comparison. Then they can.
If anybody's got any more questions, I've got a couple myself, really. There's obviously big initiatives to plant trees in Wales from the Welsh government, isn't there? As now some of these sites we're talking about might look attractive for tree planting. Do we need to kind of advocate for the waxcap grassland where that might be suggested?
Yeah, this is very worrying, because they've gone. What's going to happen is they're going to pay farmers to plant trees, and the farmers who are businessmen essentially, they're going to go, which is the less productive land, and that's going to be like the Freed type habitats, and that's where they're going to pop the trees on. Even my university's doing this. It's really embarrassing. They've got the farm that they were given in a will, and I've told them, I've said, that's a waxcap grassland. I don't know. I mean, they haven't done anything yet, but I don't get any impression they're particularly listening to me. On the plus side, even if they plant the trees, if the trees won't grow very quickly there, and say up to about 5 or 10 years, you can go back and pull all the trees out. And if you mention that to them, that's what might happen if they didn't listen to you properly, that will kind of put them off the idea that they should be thinking about where to plant the trees. The places they should plant them are the grasslands that have already been wrecked by ploughing and reseeding. There's lots of them that are very marginal. They're not very productive, and they're the ones that should be focused, because we all think, increasing tree cover is a good thing, but not at the expense of other biodiversity. The other point I'd mention, which they always talk about carbon capture, but the layperson thinks the carbon is what you see, whereas the great majority, about 70% of the carbon in a woodland or a grassland, there's more below ground than there is above ground. And especially in the unploughed ancient grasslands, the organic matter levels can be quite high. And if you plant trees on them, you're not going to increase the soil organic carbon level very much at all. And you risk decreasing it, because soil, the roots of trees tend to have a drying effect on soils, and that will then allow more oxygen in, which would oxidize the organic carbon. So you could make things worse. You could end up doing the direct opposite of what you were trying to do in terms of carbon sequestration. So that's because I think the trouble here is that Natural Resources Wales includes the old CCW people, the Forestry Commission people. I think the Forestry Commission people, they're obviously keen on trees, but they do need to think about not doing harm to other habitats as well.
Yeah.
That's my piece. I've had lots of discussions. I did a project for the Peak District, where they have similar great concerns as well about this. I think all the forestry sites that they sent me samples of them, and almost all of them had important waxcap populations on them. But there are these ploughed and reseeded grasslands on steep slopes, on uplands, that aren't very productive, and they're the ones that would benefit. You could improve things by planting trees there if you plant the right type of tree.
Fair enough.
Which is a separate worry that I'm not getting into.
Fair enough. Okay. Well, that's great. Thank you very much. Anybody else got any other comments? Any kind of questions? I can't see any hands up, I'm afraid. One thing you mentioned, this guide to wax caps in West Wales, David Bortman.
Oh no, David Harris.
David Harris.
Yeah.
You couldn't give me the details of that, or put them in chat or something, so I could actually maybe get in touch with him, maybe, and see if I can get hold of some of those. That'd be great if you could.
Let me see if I can find. If I read his email out, I don't think he'll mind me doing that.
Sure, I'll quote you. That's okay. Or anybody else could actually, if you wanted to, if you put it in.
djh.summerton @gmail.com.
Say that again.
djh.
Yeah.
.summerton.
That's SOMERTON. Yeah.
@gmail.com. That's his David James Harris, his initials. He lives at Summerton Farm, which is he was born there. So when he retired, he used to work at the refinery, Texaco refinery, and then when he retired, he went back to farming, and he then became sidetracked by fungi.
Okay.
He farms extensively there. He finds lots of new species on lots of interesting species on his farm, because he's managed it in the right way.
Yeah, and it's really interesting. What Andrew Graham's set up too was really interesting as well, because we know Andrew from moth trapping with moth trapping. Yeah. So he's a guy to go to for that. So yeah, that's really interesting to see the results of that, really. Yeah, and we're going to try it on our own plots, and maybe quite a few people maybe will get, even if how big or small their plot might be, it might be worthwhile if it's unimproved grassland. There's a possibility of managing it differently in order to encourage. So basically, if you've got a meadow, you could let the wildflowers grow as you would in a meadow, and then cut the end of July with a scythe, and then keep it low 3 centimeters for the rest of the season, and that would be the best tactic.
That would be standard that be standard practice? Well, Chris would know. That would be the
Yeah.
standard practice for a grassland site for orchids, I think.
Yes, yeah. Which of course, the orchids rely on the mycelium from various fungi, don't they?
Yeah, they seem to. I keep on thinking about this. This would be nice to find sites with it, to look at where you had orchids and wax caps and see if there was a particular association. I think, yeah, if orchids and wax caps connected with each other, that'd be really interesting.
So there's no direct connection as yet.
I don't know. You know, we get if you dig up a sward, take a bit of soil out and then pull out the roots. It's near impossible to work out what plant they belong to.
Yeah, fair enough. Okay.
So what you're describing is more or less hay meadow type management, isn't it? Like, but rather than the aftermath grazing that you might do in a hay meadow, you're cutting it again, you know, and depending on the scale, you can use sort of a scythe or some sort of mower or something like that to get the desired effect.
Yes.
It's keeping the sward short after the initial hay cut, really, isn't it? So,
I forgot one important thing, which that experiment at Andrew's farm. I'm still beating myself up 15 years later that I didn't do a plot where I cut the grass and left the clippings. I think that's very destructive doing that.
Yeah, yes.
I should have pointed that out. I'm annoyed that I didn't do that experiment then.
Yeah.
Because essentially you're not taking. I think because we have airborne pollution of nitrogen anyway, we suffer this from intensive agriculture around the place. So this falls down all over the place at quite high levels. But if you take some offtake off, via taking the clippings away or the sheep, that's helpful in just keeping the levels of nutrients low and stopping them gradually creeping up.
Yeah, because that must have happened a lot in the past, mustn't it? With maybe on like grass and road verges and things like that, and other grassy areas, because they didn't used to remove the clippings, did they? At one point. Yeah.
I think these things. The other hopeful thing, people are always worried. This might be helpful for Chris. Is there's I don't suppose many of you know Radyr in Cardiff. I think it's in North. It's where the Taff does it's a little bend in the river before it goes into Cardiff and out past the Millennium Stadium and so forth. But that's the Radyr Cricket Club is there. But that used to be horse grazed a long time ago, and then about 20 years ago, so there was some problem with the grazier, so now it's not getting grazed very much at all. And there's also, because it's next to the river, lots of Japanese knotweed invasion. There's quite large areas there, and NRW were worried about that. But when we looked at the soil in the core underneath the Japanese knotweed, we didn't find the percentage of waxcap mycelium was quite low, but all the species were still there. So if they removed the Japanese knotweed, at least the mycelium was already present, even though it was very unhappy. It wasn't quite dead. Right. So that's after 10 or 15 years of, if you like, neglect or the successional process going the wrong way. So I thought that was a very hopeful story. I thought, because they can have a campaign, they can take the half hectare of knotweed away, impose the correct management, and they're going back in the right direction then again.
Okay, well, on that note, Gareth, the hopeful story. We all hope. We hope for the best for the wax caps in Wales, considering it's a bit of a hotspot for wax caps. Hopefully you can keep it that way. I thank you very much for coming and getting, and a lot of food for thought. We're going to take away a lot with us, I'm sure, and hopefully people will look out for these things as much as they can. It's so